Hi, my name is Martha D. and I'm a graduate student at the University of Notre Dame in the Biological Sciences Department. I'm pleased to have this beautiful day here at ND-LEEF to tell you a little bit about my research that I conduct throughout the summer. Let's get started. There are many things that move within the water of a stream or river. Those could be bugs, or a piece of particulate, or the sediment from off the bottom of the stream bed. However, there are many things that we also cannot see, such as nutrients, or the focus of my research, dissolved gases. These form in tiny little bubbles that are not visible to the human eye, but play a very important role in the biological functioning of a stream. So we want to know how these gases can be created at one point, and then how far do they travel downstream before they are eventually emitted into the atmosphere. We do this in a very particular way so that we can actively study how gases are transported along these streams that have different substrates or bottom. In order to do this, I bubble argon into the top of the stream. I use argon because it is a conservative gas. That is, microbes don't use it in any way. So if I bubble it into the top of the stream, I would imagine that it would move downstream without being used by any organism. So if any of it leaves, it's because it's emitted to the atmosphere. In order to capture gas as it evades the system, we set up sampling stations at uniform distances along the stream length. When it comes to sampling for gases, it's really very simple. I've made this passive gas sampler with just a PVC pipe and some flexible tubing that serves as my outlet for my water. I have a little rubber stopper that fits perfectly into the bottom of my PVC tubing. When I'm ready to sample, I place my tube into the water parallel to flow. When I feel confident that the water that goes through is fresh and has not been interrupted by me putting the tube into the water, I use my stopper and place it at the downstream end of my tube, stopping the flow of water through the tube. I press it in tightly to make sure I don't have leaks, and then gently bring my PVC tubing up, being sure not to create any disturbance within the water itself that I've captured. You'll see that my tube is ready to go. And at this little tiny 12 milliliter vial, I place the tubing at the very bottom, pinching the tubing to restrict some of the water flow. I allow enough water to refill this vial 3 times over. That is to ensure that I have captured the water that has the least amount of interaction with the atmosphere, or I'm getting the purest sample as I would have found from the stream itself. Once I feel confident that it is overflowed, I bring my water up, I can dump my excess, and I seal it with this airtight cap that actually has a layer of Teflon on one side and butyl rubber on the other side. I am able to just screw that on like any other cap, but it creates an air and watertight seal. If I've taken a correct sample, there should be no bubbles within this little vial. After I've brought these back to the lab, I run them or analyze them on a membrane inlet mass spectrometer. We call it MIMS for short. This machine directly samples the water and draws from it the argon, which as you'll remember is what I'm injecting into the stream, as well as oxygen and N2, or a primary component of our atmosphere. From there, I am able to see how much argon I captured in each one of my samples. That said, we would imagine that 5 meters down, we'd have the most amount of argon. Compared to 50 meters down, we may still capture some argon, but it's relatively less than what we capture upstream. That's because this water is flowing over these rocks, getting caught in the little crevices, and oftentimes emitting out to the atmosphere. The purpose of this study is to apply what we find in these small experimental streams to larger rivers that are more difficult to sample. Thanks for joining me here at ND-LEEF. I'm so happy that I got to spend a little time showing you what my research is all about. Until next time.